Hello, this is Ethan. I am reviewing article number 6. This article explores the feasibility of using colonic organoids derived from human-induced pluripotent stem cells on modeling colorectal cancer and drug testing. So here's an overview of what we'll go over in these subsequent videos. First, I'll start off by giving some context and background to the study. Some of the following information has been covered in previous modules, but they are relevant enough here to warrant a revisit. So I apologize in advance if any of it's overly redundant. I'll also discuss the results and eventually the overall study, the importance of the data, what this means for future studies, and also why and how the study could be improved. Okay, so this study focuses and utilizes cells from specific portions of the endodome. And just so it's clear to everyone, I'll quickly go over what the study refers to when it discusses definitive endodome, hindgut endodome, or colonic organoids. Here's a simplified view referred to as the gut tube. In a nutshell, the paper uses cells from the foregut and the hindgut. The hindgut refers to a specifically vascularized region of the gut tube. As you can see here, all these regions have like their own arterial supply. The hindgut refers to the specifically vascularized region, which has the colon and everything else there. The foregut has the gallbladder, the liver, stomach, pancreas, spleen. And another thing that we'll try to remember here is anterior is up, or closer to the mouth. Posterior is closer to the behind. Because they'll take sections from two distinct points here. Okay, so the hindgut comprises the posterior-most one-third of the system. This may further be divided into the small intestine, large intestines, and essentially the rectum. This study will label the iPSCs samples from these specific subdivisions. Okay, so here's another thing that the study relies extensively on. The Wnt beta-catenin signaling. You might remember this from module 10. So the experiments throughout will measure Wnt signaling either from the point of view of the APC, or they'll take measurements from a couple of proteins off the Wnt target genes, kind of just measuring the products rather than one of the internal messengers. Essentially, the authors are covering all the bases in regards to Wnt signaling. It's important to frame Wnt signaling in regards to two aspects. By measuring APC levels, Wnt signaling can be measured as on or off. So a couple of the experiments in the paper will compare APC levels between wild type and experimental levels as indicative of proliferation. And secondly, the products of this signaling will only show up if Wnt is carried through, so it's used as further evidence toward the levels of Wnt signaling. So the author's objective here is to develop an effective protocol for deriving colonic organoids from differentiated human embryonic stem cells, or iPSCs, for the purpose of modeling human disease of the large intestine. And this paper describes a strategy for driving human colon organoids. This process can be extended to FAP iPSCs to develop disease in a dish platforms to monitor the overactivation of Wnt signaling associated with too much proliferation of the epithelial cells of the colon, which is pretty relevant to colorectal cancer. And that's the background of the paper, so let's go into analyzing the figures.